All right, let's dive in. Today, we're going to journey into a universe that's completely hidden from the naked eye, one that exists on a scale that is honestly hard to even wrap your head around. We're talking about the building blocks of all life, the cell.
I mean, just stop and think about that for a second. Right now, on the tip of your finger, there is an entire world bustling with life, a whole ecosystem, and you can't see any of it. So the question is, how do we even begin to peek into that world? You know, for most of human history, that hidden world was just an idea, pure speculation. But then, we invented a key. And that key, the microscope, didn't just unlock a new world, it completely changed our understanding of life itself.
And the crazy part is how quickly it all happened. It really started back in the 1590s with a guy named Zacharias Janssen, who was just messing around putting lenses in tubes. Then, only a few decades later, Robert Hooke looks at a piece of cork and sees these tiny, box-like structures. He calls them cells. And then Anton von Leeuwenhoek comes along and builds this incredible device that could magnify things 275 times, revealing for the first time ever that there were tiny living things swimming around everywhere.
So, all of that history leads us right to the tools we have now. You've probably used a light microscope in science class, and they're amazing. But to see the really, really fine details of what's going on inside a cell, we need to call in its much, much more powerful cousin, the electron microscope. And we'll get to that in a bit.
First, though, let's talk about the classic light microscope. Using one is actually a pretty methodical process. You always start with the lowest power lens to find what you're looking for. Once you've got your slide on the stage, you use that big knob, the coarse focus, to get a rough image. Then, and only then, do you switch up to the higher power and use the little knob, the fine focus, to get everything perfectly sharp.
Okay, so you've got it in focus, but here's the catch. Most cells are basically transparent. They're see-through. So to fix that, scientists use stains. Just a tiny drop of iodine on some onion cells or methylene blue on your own cheek cells, and suddenly, bam, all these internal parts, like the nucleus, just pop right out. It's kind of like turning up the contrast on a photo to see all the hidden details.
So now that we have our tools and we have our techniques, let's actually start our tour. And we're going to begin with the complex cells that make up plants, animals, and yep, us.
The big, fancy name for these is eukaryotic cells. And the most important thing to remember about them is that they have a true nucleus. Think of it like a secure vault, a special compartment that holds all the genetic blueprints, all the DNA, safe and sound.
Now, within this eukaryotic family, you've got your two main players, animal cells and plant cells. They share a ton of the same core machinery. But plant cells have three extra super important features: a tough, rigid cell wall for support, these things called chloroplasts to do photosynthesis, and a huge permanent vacuole, which is kind of like a water tank that keeps the cell firm.
And this is where that whole city inside a cell idea really comes alive. The nucleus, that's City Hall, calling all the shots. The cytoplasm is the jelly-like stuff where all the action happens. The cell membrane is like the city's border patrol, controlling everything that goes in or out. The mitochondria are the power plants, generating energy for the whole operation. And the ribosomes? Those are the tiny little factories, just churning out proteins all day long.
Okay, so that's the complex stuff. But what happens when we zoom in even further, like way further? Let's explore the much simpler, much more ancient forms of life, like bacteria.
Bacteria are what we call prokaryotic cells. And that little prefix pro is the whole key here. It literally means before. These are cells that evolved before the nucleus. So their DNA, their genetic blueprint, isn't locked away in a vault. It's just kind of floating around loose in the cytoplasm.
And when you put them side by side, the difference is just whoa. First off, size. It's not even close. Prokaryotes are way, way smaller. They're also so much simpler inside. I mean, eukaryotes are jam-packed with all sorts of specialized parts, but prokaryotes, they basically just got ribosomes and cytoplasm. No nucleus, no mitochondria. They're these super lean, mean, efficient survival machines.
So that brings up a pretty big problem, right? If a regular light microscope is already pushing its limits to show us the details inside a big eukaryotic cell, how in the world are we supposed to see the even tinier structures inside a bacterium?
The answer isn't just about making things bigger, it's about something called resolution. Resolution is all about clarity, about seeing fine detail. Think about it like this. You can take a low-quality photo on your phone and zoom in, right? You're making it bigger, but at some point it just turns into a blurry, pixelated mess. You're not getting any more information. It's the exact same idea with microscopes.
And this is precisely where the light microscope hits a wall. Its best possible resolution is around 200 nanometers. Anything closer together than that just blurs into one blob. But the electron microscope? Its resolution is less than 1 nanometer. That's not just an improvement, that is a total game changer.
So how does it do that? Well, the secret is right there in the name. Instead of using waves of light to see something, it shoots a focused beam of tiny particles, electrons. And that's what gives it this unbelievable resolution, letting us see things we could only dream of before, like individual ribosomes or even viruses.
Okay, so now that we can finally see this world, we kind of need a new way to measure it. So let's talk about the math for a second, because the sense of scale here is just mind-blowing.
First, the basic formula. It's actually super simple. If you've got a drawing of a cell in a book that's, say, 10 millimeters wide, and you know the real cell is only 0.01 millimeters wide, you just divide the image size by the real size to get the magnification.
But with an electron microscope, the magnification is just ridiculous. A good transmission electron microscope, or TEM, can magnify an image about 1 million times. Let that sink in. 1 million. That's like taking a single grain of sand and blowing it up to the size of a tall building.
To even talk about these tiny sizes, we need a whole new set of measurements. The key thing to get here is that each step down the ladder, from a millimeter to a micrometer, then from a micrometer to a nanometer, is a leap of 1,000 times. A nanometer is just, it's 1/1000th of a meter. It's almost impossible to picture.
And this quote just nails it, doesn't it? A light microscope can't tell two things apart if they're closer than 200 nanometers. A powerful electron microscope can tell two things apart that are less than 1 nanometer from each other. That's the difference between seeing a city from an airplane versus being able to read the license plate on a car driving down one of its streets.
So, wow. Let's just take a breath and think about the journey we just took. We went from the very first simple lenses all the way to powerful electron beams. We've toured the complex cities inside plant and animal cells, and we've explored the stripped-down, efficient world of bacteria. We've seen a whole universe that's working on a scale that's almost too small to imagine.
And really, that's the most incredible part. Every single living thing, you, me, the tree outside, is a universe built from trillions of these unbelievably tiny, complex structures. And it just makes you wonder, doesn't it? With the tools we have now and the ones we're going to invent tomorrow, what else is out there just hiding in plain sight?